The overall goal of this experimental protocol is to enable inexperienced PTM investigators to effectively determine if their target protein is endogenously modified by one or more PTMs using a single comprehensive system.
This method will enhance investigation in the post-translational modification field by enabling detection of key PTMs and potential crosstalk for any target protein using a single simplified system. The main advantage of this technique is that non-PTM experts can detect whether a protein is endogenously modified by one or more PTMs in their model system.
We first had the idea for this method when studying regulatory mechanisms for several cell signaling pathways and discovered that user-friendly kits or systems were not readily available for studying non-phospho PTMs.
Begin this procedure with preparation of A431 cells as described in the text protocol. Also prepare blaster lysis and dilution buffers with inhibitors. Pour off the culture media and place the cells on ice. Then, wash the cells twice with 10 ml of 1x phosphate buffered saline. Remove as much PBS as possible prior to adding blaster lysis buffer to maximize cell lysis. Add 600 microliters of blaster lysis buffer to each 150 mm plate. The amount of buffer is based on expected protein yield. Next, lyse the cells using a cell scraper. The lysate will become viscous due to nuclear lysis. Use a snipped 1 mm pipette tip to transfer the crude lysate into a blaster filter that has been placed in a 15 ml collection tube. Use a supplied filter plunger.